Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. I am Shakil Ahmad Mehman and I am presenting you the description of the carbohydrate. We have discussed the monosaccharide as well as disaccharide. Disaccharide is among the oligosaccharides. They have the only two monosaccharide units, whereas the oligosaccharides, they have the 3 to 10 monosaccharide units. They are linked together with the monosaccharide groups by the glycosidic bonds. Oligosaccharides, they are branched or unbranched. It means their chemical structure may be branched or unbranched. Unbranched means it has a linear sequence. They are in a line. They must be in a line. Whereas the branched, they are been connected from the linear towards the branches. Therefore, they are branched and unbranched. Dietary oligosaccharides source is that whenever we discuss in a way, they cannot be digested by the human body. Because of that, they have the no calorific values. Oligosaccharides are the components of the glycoproteins. So this is all about the oligosaccharides. Now we move the polysaccharides. We can say these are the glycans. Polysaccharides are the simple glycans, consist of the repeat unit of the monosaccharides or their derivatives held together by glycosidic bonds. They are primarily concerned with the two important functions and storage of the energy. Polysaccharides are the linear as well as the branched polymers. These are the monosaccharides. They have the more than 10 monosaccharides. They must be 10, more than 10, as well as many monosaccharides are there. This is the contrast structure of the protein and nucleic acid, which are only linear polymers. We can see here as well as this is the polysaccharide complex carbohydrate. They are the same and different monosaccharide units and their derivatives repeatedly linked by the glycosidic bonds, whereas we can see their general formula where N is denoting the number of the molecules as well. They can be identified by the test as a characteristic test of the polysaccharides. We can see iodine test. We can identify where the polysaccharides are present by the heating process of the collation. The occurrence of branched and polysaccharide is due to the fact the glycosidic linkage can be formed at any one of the hydroxyl group of monosaccharide. Whereas we move forward, the polysaccharides are the polysaccharides. These are the two types of the polysaccharides, homopolysaccharides and heteropolysaccharides. Whereas the homopolysaccharides on hydrolysis, only a single type of monosaccharides are attached together, unite together. They are named based on the nature of the monosaccharides. Thus, glucans are polymers of the glucose, whereas the fructosanes are polymers of the fructose. The simplest way to identify is that glucagon, this is the derivative of the glucose, whereas you can see the fructose is another polymer of the fructose is the fructosan. On the other side, we can say the heteropolysaccharide on the hydrolysis yield a mixture of few monosaccharide or their derivatives. There are few examples of the glycosans. This is the also from the polymers of the glucose. You can see here the examples of the glycosans: starch, glycogen, cellulose, dextrin and dextran, whereas on other hand the fructosan. Fructosan is the type of the homopolysaccharides. Repeating unit of the fructosan is the fructose. The fructosan is a polymer of the fructose. This is the example of the inulin. Now we move the glucosan, that is the example of one of the example of the starch. Basically, starch is a carbohydrate reserve of the plant, which is the most important dietary source of the higher animal, including man. High content of starch is found in cereals, roots, tubular vegetables. Starch is homopolymer, composed of the D-glucose unit. It means this is the glycosan, and glycosan is the polymer of the glucose. That's why it is composed of the D-glucose. You can see here the starch is the glucagon, repeating units of the alpha D-glucose, and it has about the more than 7,000 monosaccharides group glucose units. Component of the starch is basically, it is composed of the two main things, amylose and amylopectin, whereas the amylose is about 15 to 20% of the total starch, and amylopectin is the 80 to 85%. You can see here, simple as that. The amylose is basically is composed by the linear chain. It is not branched, but it is the linear chain, and it is composed of the D-glucose linked by the alpha at anomeric carbon 1 to 4 glycosidic bond. We move forward, we can see here the structure where we can see the amylose is a linear chain, a straight chain is there, whereas when we compare the amylopectin, it is the branched, unbranched. Amylose is the unbranched, whereas the amylopectin is the branched. It's composed of the starch about 80 to 85%. It means starch is have two components, amylose and amylopectin. Amylose is unbranched, whereas amylopectin is branched. Amylose contains 15 to 20% of the total starch, whereas amylopectin composed 80 to 85% of starch. Branched structure, it appears every 20 to 30 glucose units it has a branched. So therefore it is connected with each other by the glycosidic bond and the anomeric carbonate 1 to 6 point. And whereas the glycosidic bond is a linear, where the linear is there, that is the alpha 1 to 4. Now we move forward to see the structure of these amylose. This is the basically amylose is the monomer. This is the sequential straightforward, as amylopectin is the branch. There is A and B. You can see A is the linear. They are the connected with the 1 to 4. That is also seen very well. And whereas you can see here, you can see here as well, this is the A, this is the number B. You can see there is a linear chain 1 to 4 anomeric bonds, and whereas the branch, it is amylopectin that is connected with the 1 to 6. Potato is the main source of the starch. This is the amylopectin. This is the branched structure of the amylopectin. Starch is basically white odorless tasteless powder. You can see starch insoluble in a water, but it is solubilized in the heating. Whenever we heat up this one, that can be converted by the gel and paste form. This is the non-reducing carbohydrate starch and negative Benedict taste, whereas as I earlier said, the iodinic reaction can give you the blue color and specify the polysaccharide is present. Occurrences is there: plant, seed, tubers, roots, raw fruit. Dietary source is the grain, rice, wheat, jawar, bajra, potato, etc. and etc. You can see in this. Biomedical importance is there. Starch is the storage from the glucose. As I earlier said you, it is the reservoir energy in the plants. It is very useful source for the dietary useful source for the animals, including man. It is ingested and hydrolyzes by the salivary glands as well as pancreatic enzymes. Digestion, you can see amylase by the pancreatic, alpha pancreatic and salivary gland amylase enzyme is there. It is break up by the heating up at the 1 to 4 anomeric bond. It can be easily break, hydrolysis and converted into the maltose. Maltose converted into the isomaltose. Then it is converted into the glucose, small unit of the glucose, and that will gives you the more energy source to your body. This is all about the starch. Now move forward the glycogen. It is also one of the example of the glycogen. As you know, this is the oligosaccharide as well, and it is the homopolysaccharides. Glycogen is found in the animal homopolysaccharides, animal starch. We can say this is the animal. Glycogen is a carbohydrate reserve in the animals. Starch is the reserve into the plants, whereas the glycogen is the reserve into the animal. In present high concentration in liver, followed by the muscles, brain. Glycogen also found in plants that do not possess the chlorophyll, yeast or fungi. We move forward. This is the chemistry of the glycogen. Glycogen is chemically glucosine, repeating units of the alpha D-glucose, and these have the 20 to 30,000 glucose units. You can see here, this glycogen is the amylopectin structure. Like amylopectin, it means it is the branch. Amylopectin is the branch. Branching pointing appear after every 8 to 10 glucose units. They are linked together with the glycosidic bond, but after every glucose unit they connected with the branch structure at alpha 1 to 6 anomeric carbon. Linear structure has glucose linked, they have the as well as the linear structure, they add same as the starch 1 to 4. This is the structure of the glycogen, whereas it is present, you can see here 1 to 4 and 1 to 6. This is the branch connected in the branch. This is the linear one, the structure of that. You can see here these are the green points are the branch structure of that, where red are the linear structure is there. We move forward. Glycogen has more at alpha 1 to 6 branches. Highly branched structure permits rapid glucose release from the glycogen stores. As I earlier said, it is stored in the liver. Whenever the extra, it is stored into the muscles as well as the brain. At the center of the glycogen molecule lies glycogenin, protein premier of the glycogen synthesis. Structure of the glycogen is similar that, as I earlier said you, the amylopectin with more number of the branches. So we can now further move towards the presentation in that the dietary source, where is the present in animal, as I already discussed, and for the storage you can see the diet source of the dietary glycogen is the human being, which has high dietary and calorific values, whereas the starch has the no calorific value in human being. After well-fed condition, free and excess of the glucose is the condensed to the glycogen and glyconeogenesis. The main source where it stores it is the liver. By the synthesis process of the glycogenesis, whenever it needs, it will be a breakup in the glycogenolysis, or you can say glycolysis, that is required for the energy as well. Broken up, you can see this one is broken up the glyconeogenesis whenever the body is required. The biomedical importance storage in glucose, human being as well as animal body after the fed condition, it serves as the reservoir the glucose which can be used in emergency condition or between two meals. When we are, for example, taking the breakfast, after that we will take the lunch. Between two meals we need energy that will provided by this stored energy from the liver, and if required more, if you are getting hectic, if you are doing exercise, if the reservoir is finished in the liver, then it comes from the muscles as well. A condensed form, this is the condensed form, minimize the isometric effect and occupy less space. It is broken, very easily broken into the glucose. So glycogen produce less isometric pressure and occupy small space. Glycogen is the soluble in water. Iodinic reaction, we can identify the glycogen is present. The deep red color will provided by this. Dietary GI in gastrointestinal tract, this is the digested by the alpha amylase to maltose and isomaltose, and finally thousands of the glucose units. So it will provide us energy by the glucose. Liver is the glycogenolysis in human body, regulate the blood glucose level in fasting condition between two meals. Muscles as well as glycogenolysis, this is also provided from the another source is the muscles. Now we move forward the other glucosan example. This is also homopolysaccharides. This is the very much rich source, extensively, exclusively provided into the plants, and it is the most abundant organic substances in the plant kingdom. It is the predominant constituent of the plant cell wall. Cellulose is totally absent in the animal body. Cellulose is composed of the B-D-glucose units linked together by the 1 to 4, we can see here as well. Repeating units are there. They are linear. They can be unbranched. Structure where beta D-glucose unit repeatedly linked by the beta 1 to 4 anomeric carbon glycosidic bonds. We move forward into that. They can be, you can say easily, they cannot be digested by the human, and they are abundant carbohydrate, plant cell wall, as I earlier discussed, dietary rich source in cellulose, whole grains, green leafy vegetable, cabbages, legumes, nuts, beans, dates, and other as well, etc. Biomedical importance is that cell wall structure is the mechanical support to the plant leaf as well. Wood, cotton, paper composed primarily with the cellulose, and human dietary cellulose is not digested. You can see the bond amylase is break, and alpha bond only certain ruminants. You can see the cattles, they only give you the dietary cellulose. In humans serves as dietary fibers. Whenever we are taking it, they are usually used as the fiber food. But we can compare into the animals. They are certainly ruminants and herbivorous animal, certain microorganisms in the gut which produce enzymes that can cleave beta glycosidic bond, that is broken down, and hydrolysis of the cellulose yield is disaccharide cellobiose followed by the beta D-glucose. Cellulose, though not digested, has a great importance in human nutrition. It is the major constituent of the fibers. Non-digestible carbohydrate, we can say, increase intraluminar pressure, reduce the transmit time of the feces to remain in gut, eliminate daily metabolic waste and toxins out of the body through feces, or defecation with the greater ease and good frequency. Cellulose prevents constipation and reduces the risk of the colon cancer and varicose veins. Varicose veins is the you can see there is a problem in the leg vein as well, another vein that also gives to the blood towards the heart. Diverticulosis of intestine. Diverticulosis of intestine is the basically of the swelling, hemorrhoids as well as a swell up from your rectum. Cellulose reduces the absorption, glucose, cholesterol, and ameliorated the condition of the diabetes mellitus, atherosclerosis. They can prevent from this as well. Cellulose being non-calorific and possessing high satiety value help in the managing obesity. Because of that, it is the fiber food. You can see for maintenance of the good ingest dietary fiber, RDA for the dietary fiber, main good health is the adult 20 to 25 grams per day, children 5 to 10 grams per day. And the disadvantages of this is the dietary cellulose decrease the absorption of the minerals. Now we move towards the